So in this video, I'm going to talk about enzymes and how they work. And again, I want to remind us of the main things we're going to be focusing on with these models. Again, don't get too bogged down in the details, right? You want to understand these diagrams. You don't need to memorize how they look, right? The main things we want to look at are structure and function. Structure and function. We want to look at what I call the critical steps. This is not, I don't think, a technical biological term, but it's what I consider them. They're the steps where the process is most vulnerable, right? And then we want to look at regulation. And as I mentioned, this may not be on the AP. It still could be, but I doubt it would be. But I also do want to focus on how, or at least mention how this relates to fitness. Fitness. Okay. So to start, right, enzymes, and we have the protein structures, right? Enzymes are proteins. It's proteins that help speed up reactions and lower activation energy, and I'll get to that in a second. But I want to talk about the main structure of these enzymes, which are in proteins in general. They have four structures. They're the primary structure, and what these are just chains of amino acids, right? They're the basic building blocks of proteins. They chain together. When you have a large chain when you have a large chain of amino acids, they end up folding or in some way. So sometimes they'll make a let's see, it's like a sort of squiggly, like a curly Q, right? Like it sort of looks like a curly fry in a way. Or they could make like the folded sheet pattern. It doesn't, it's not super important to know these specifically. What's important is that when you have a bunch of amino acids, because of how they interact chemically, right? Some are nonpolar, some are polar. They all have different properties. They form different structures, right? And so then these secondary structures, which are made up of like folded patterns of amino acids, those combine. You have a bunch of secondary structures, as you can sort of see in here, that form a tertiary structure, which will look sort of like a protein. But then a bunch of tertiary structures fit together, right? These are a lot of the time separate chains, right? This is just all one. These are sort of, in a way, the tertiary structures, all one chain of amino acids that have different sections that fold in different ways. But then they fit all together and have a bunch of these protein structures that fit together to make one big protein, the quaternary structure. Right? So this goes to structure and function, right? How does this work? Why is this physical structure of the protein so important? It's because of the lock and key model. What that is, and here I have an example of of a substrate, some material fitting right into the enzyme, and the enzyme keeps it together and helps it, because of its structure, helps it break apart into two. Enzymes can also do that where they take two things and put them together in one, right? Or there's a bunch of different things they can do. This is just an example with a decomposition reaction as opposed to the other way, which would be a synthesis reaction. And so the substrate goes in there and it breaks apart with the help of the enzyme. And I want to talk about what the enzyme does quickly. It does two main things. It speeds up the reaction, right? And then it also lowers activation energy. Lowers activation energy. And what that is that's just the energy that the process requires to just get started, right? And again, this does also relate to fitness, right? Just going sort of back and forth, going to fitness. if you have enzymes, what it lets you do is it lets you do more things, more reactions, in a shorter amount of time with less energy, right? So that will obviously be an advantage for an organism. Okay, so back to the structure and function, right? It fits right together. So this structure, the enzyme, is incredibly important to how it works together, because if the structure if everything didn't fit together perfectly to make the exact right shape, it wouldn't be able to bind to the substrate, right? Which brings us to our critical step. Where is sort of the easiest place for this to go wrong? And I might argue that the most critical part of this, right, where the place where it's really easy to go wrong, is right here in the primary structure. And this actually happens in real life with certain proteins, that if you even exchange just one amino acid different. Sometimes it will be like exchanging a polar and a nonpolar amino acid, or other types. I think there's some hydrophilic amino acids, or charged and non-charged, right? So there's a lot of different types of amino acids that behave differently in terms of, forming these structures. If you just switch out one amino acid, you could end up messing up just one of the secondary structures, because it will, maybe get repelled from another amino acid, or just end up folding differently, because each amino acid has different chemical properties. Right? So switching out one amino acid in this primary structure could mess up the secondary structure. And then all the secondary structures may not fit together perfectly into the tertiary structure, which would mean that this tertiary structure might not fit into the quaternary structure, right? So just messing with one amino acid, switching one amino acid, not always, but sometimes, this could end up having a huge effect on the quaternary structure, to the point that the protein couldn't actually work. Right? So that's kind of incredible, really, that one amino acid different could mess up the whole protein. Okay. There's one other thing that could mess with the structure of the protein, right? Which is temperature and pH, right? The acidity can mess with the structure of the protein, right? They can change how the amino acids bond, or they can affect the amino acids. So if you have a different temperature or a different pH, sometimes what that will do is it will be called, it's denaturing the protein, right? Denaturing the protein, what that will do is change its shape so that it can't do this. So what can these can denature. Okay, they can denature proteins. Okay. So next, right? Let's look at the regulation aspect, sort of the last aspect. And I have three different forms of regulation here. I've got competitive inhibition, non-competitive inhibition, which is also called allosteric inhibition. I have a line connecting these. These are the same. And then allosteric activation. These are all forms of regulating enzyme activity. Okay, so let's look at competitive inhibition. What this is you have an inhibitor, that's this purple thing over here, that what it does is it binds to exactly where the substrate is supposed to bind to. And it is clearly blocking the substrate from binding, right? That is called competitive inhibition. That is one form of, right, if you don't want an enzyme to work, you just release a lot of competitive inhibitors, right? And that makes the enzyme sort of stop working, right? It competes. This one works very well when there's a lot of competitive inhibitors. So say you want a process to shut off if you have a lot of something, if you have a high concentration of something, right? You'll use competitive inhibition, right? Because when it's competing with the substrate, if there's not a lot of inhibitors, then the process will still go on, right? Because there needs to be essentially more inhibitors than substrates in order for for it to win, because they're competing, right? So that's one way that an organism will use inhibition. Say if they like require a lot of something. Now, what if they just want the inhibition to happen even with just a little bit, right? You don't want them competing. You don't want there have to be more inhibitors than substrates, right? Some processes might want that, some won't. Then you use non-competitive inhibition, which is you have what is called an allosteric site. You don't need to know that term, but you may see it on the AP exam. And what happens when you have a non-competitive inhibitor bind to the allosteric site is it actually changes the structure of the protein. It can actually do two things. It could either stop the protein from helping the reaction go forward, or actually change the protein structure, right? So you sort of see that in both. Here it says substrate can bind but reaction is blocked, but sometimes the inhibitor could just stop the substrate from binding in general. It could change the shape. It's called a conformational change, right? And so that will happen sometimes when you have an allosteric inhibition. The last type is allosteric activation, right? Where instead of inhibition, you're not saying that you want to send something in to stop the reaction, but you're saying that this reaction can only happen if it's turned on. And so what you have is you have an activator, instead of an inhibitor, that binds to the allosteric site, goes through a change, but this change, instead of blocking the substrate, it changes the shape. It goes through, the protein goes through a conformational change, and it allows the substrate to bind, right? So an organism really has sort of a pick based on, how evolution works and how, different processes you want them to be regulated. Some will prefer you have to have a lot of inhibitors so they compete with the substrates. Some will have you want to release something that will just turn it off. It doesn't have to compete. That's non-competitive or allosteric inhibition. And then allosteric activation, when you basically send a signal telling the protein to start working. Right. And so that's essentially proteins. The only other thing I want to talk about is protein activity, right? Based on another way of regulation, is the amount of substrates, right? And in fact, the amount of substrates isn't going to make a huge difference. That's actually a big misconception. The amount of substrates aren't going to make a huge difference in in how well the proteins work. What will make a difference is temperature, pH, and the amount of proteins. So that's just an important thing. I doubt the AP will test you on that, because that's, again, sort of like, just recalling facts. But the amount of substrates, right? The amount of substrates has no effect. Or at least almost no effect. I mean, if you have very few substrates, if you have like very, very few substrates, then maybe it will have an effect. What really will affect are the amount of proteins. Big effect. Right? You still need to have substrates, but having, a lot versus like just a lot, no effect. But the more proteins you have, the more this process works, right? And then the other ways to regulate it, are by having activators or inhibitors, or by regulating temperature and pH and acidity. And then, of course, if you have more substrates, that's not a form of regulating this protein's activity.